All right, welcome back. So intrinsic factors, that is the topic for today. As mentioned in the last video, so these are basically the things that we are kind of inherent to a food product, or maybe some things that we can add to a food product that will kind of minimize the growth of undesirable microorganisms. So before we jump into that, wanted to tell you a little bit of a story here. So let me start by sharing my screen here. So since, sometimes, with some of these classes, I do like to at least start off a little bit, telling, about some, story that is kind of relevant to, current food microbiology, or in this case, it's a story I've used before, because it was, came out of kind of a news article from 2014, as you can see up here. But it actually, I think in the age of coronavirus, it kind of comes back again, in terms of, thinking about during, infectious, times, what is the role of wearing gloves? I mean, and, a lot of this all started to happen in the spring. We heard a lot about social distancing. You started to hear about wearing masks, and there was some information that you hear out there about people would say you should wear gloves to protect yourself from coronavirus. And really, if you kind of go back and revisit the whole idea of why we wear gloves and what exactly we're trying to do, you can see that begin to see that it might not necessarily be true that wearing gloves is going to protect you against infectious diseases all the time. So where this story begins, as I said, it was back in 2014. And this was in a story that came out of a law that was working its way through California, in which I think you can appreciate on a normal year, you can go through downtown, go to your favorite fast food place, takeout place downtown, and the person who's preparing your food is wearing gloves. And there was a law that was working its way through California, in which some state legislatures wanted to pass a law in which basically say that wearing gloves during food preparation is going to be completely optional. It's going to be up to the establishment to decide whether that is the the best thing to do. And let's take a little bit of a segue away from that for just a second to talk about a document that provides a little bit of guidance to us, in terms of what exactly is required. And well, not actually, that's even going too far. Not even required, but what's recommended. If you were somebody who's starting a restaurant, you're starting a fast food place, and you want to find a document that's going to give you some information about what are some of the things that you should be thinking about in terms of procedures to put in place, employee training. This is one place that you can go. It's a document that's put out by the Food and Drug Administration called the Food Code. As you can see, 768 pages long. I'll put a link to this on, actually I'll download this to the Canvas site. Obviously, do not read this entire thing unless you really need something to put you to sleep. So what the Food Code is basically, as I said, it's guidance, and by guidance I'm really saying it's not regulatory. So by regulatory we mean information that's put out there that by law that you're going to need to follow. This is really more of just some guidance for restaurants and otherwise, as I said, for things to think about incorporating into your day-to-day, workflow. The part we're going to focus in on today, or at least for this part, let's see if I can find it really quick. I know it's actually page 65 in here, but it's information about what types of protocols, employee training do you use, to protect against contamination after you've received raw ingredients. And the first part, preventing contamination by employees, has a whole section on preventing, transfer of potentially hazardous microorganisms to foods that have been transferred there by, by your employees, using their hands. Got a whole bunch of information out here about, some guidance, including, a place that you can go to learn a little bit more about, what's the recommended way for employees to wash their hands. You'll have all sorts of exceptions, that are thrown in here, depending upon, what types of foods, you're preparing or how you're preparing it. Let's jump down to, so this section E, where I want to focus in a little bit more on, in which it's, for food employees, that you're not serving a highly susceptible population. So not immunocompromised, a large number of immunocompromised or cancer patients or people who are highly susceptible, to diseases. A number of exceptions to the rules that this, to the guidance that this document is providing, and including, if we go down to section 6 of Part E, it's basically gives you some direction for various activities that if your employees are participating in these, that it's probably can be considered acceptable for them to handle food using bare hands. What they're recommending in here is that if you are following 2 or more of the following guidelines, that you could probably find this acceptable. One of them is double hand washing, is what it sounds like. Wash your hands, rinse them, wash them again. Clearly, hand washing is something we've heard an abundance about in the last 6 months or so. The use of nail brushes, so small brushes that allow you to kind of scrub the dirt out from underneath your fingernails. Your fingernails are an excellent place for microorganisms to hang out and to eventually get transferred to food. If you're using antiseptics, again, something we've heard an awful lot about the last few months, you're using incentive programs. So this is kind of an interesting one, because this is not something that you're actively doing, of course, while you're preparing the food the way that the 3 above it are. But if you're an employer and you offer paid sick leave to encourage employees to stay home, when they're feeling ill, of course that's going to minimize the chances that a sick employee is going to come in, and is going to transfer whatever that they're carrying to a product that's being purchased by a customer, or of course the big catch-all, or you're using some other type of approved controlled measures. Or that are approved by the regulatory authority. So basically a way of saying this document is flexible and it's going to change somewhere in the future. So we already have some guidance here to tell us a little bit about when you might when it might be acceptable for employees to contact food with bare hands. So let's get back to this document and learn a little bit more about, well, what exactly were these lawmakers trying to argue? And so some of the things that, well, I guess as you say, that is kind of an advantage to wearing gloves. Is of course the idea is that you're going to protect you're going to prevent the transfer of microorganisms from your hands to the food products at a kind of an appreciable level. It could also, if you've got an employee who maybe has a skin infection or something like that, again, you're going to minimize the transfer of microorganisms. So some of these common sense things that we can see why glove wearing might be beneficial, but where this argument was coming from, and saying, well, is this really protecting public health the way that we think it would? Some of the counter arguments are, well, if you're wearing gloves for long periods of time, obviously you're building up like a warm, humid environment between your fingers and the gloves, just from the sweat of your hands, things like that. And providing heat, providing moisture, we already know from the last lecture, that's stuff that microorganisms love, so could you, especially if you're wearing gloves for an extended period of time, be creating an environment where you're actually encouraging the growth of certain microorganisms. Going off of that, gloves are not fail-safe. Say you've been wearing them for a long period of time, you've had that microbial growth, and you get a small tear in it. You might now actually transfer a greater number of bacteria, to the food than you would have if you weren't wearing gloves. Other arguments are, gloves are really good at stopping or preventing the transfer of bacteria from hands to food. They might not be as well preventing the spread of viruses that are much smaller than bacteria. And as we'll learn when we talk about viruses, at the end of the semester, there's actually more foodborne illness in this country due to viruses than there are to bacteria. But probably one of the biggest arguments that people really have about wearing gloves is not so much from the safety standpoint of do they or don't they prevent the transfer of bacteria. Clearly they do. But the idea of, does it give people a false sense of security? So basically, do people, when they're wearing gloves, do they think that by virtue of the fact that I'm wearing gloves, I'm not going to transfer any potentially virulent bacteria from myself to the person who is purchasing this food? And I think, clearly that is completely untrue. I mean, you probably have been somewhere where somebody's prepared food for you, they're wearing gloves. After they're done, they handle your money wearing their gloves. They use a cash register wearing gloves, and they're not thinking about the fact that they could be transferring bacteria from surfaces, from money onto those gloves that they eventually get into the food. So it's this idea of the false sense of security that was actually part of a study, and I'll put a link to this again on Canvas, through the Centers for Disease Control and Prevention. So our major public health agency that's in Atlanta, Georgia. They did a study back in 2006 that was looking at practices of hand washing and hand hygiene in various food preparation places. Some of the things that the study found that are shown here at the bottom, is that they were saying workers engage in about 9 activities an hour that should involve hand washing, but only 25% of those activities, do the workers actually wash their hands after performing. This list down here are, the hand washing rates, after, doing certain things, such as, before preparing food. That's an easy one. I mean, logically, you think I'm going to prepare food, I'm going to wash my hands. Well, 41%, of all individuals actually wash their hands at this point in time, and a low of 10%, just after generally, touching their body. This sentence right down here, at least to me, is a real key to this, where they say, workers were more likely to wash their hands at the right time when they were not wearing gloves than when they were. So again, I think it really speaks to that false sense of security that many people get if they don't receive the proper training in food preparation. Again, the fact that you're wearing gloves does not make you immune to transferring bacteria from one surface to a food product. So just ending that story there, and with the kind of just giving you my own personal point of view here. I mean, personally, I think wearing gloves is a good thing. Clearly, as we mentioned, and as we know, you're not. It lessens the likelihood of transferring bacteria from your hands, from cuts you may have onto food. The problem is people need to receive proper training of how to wear them, when to remove them, and just being conscious about the fact that wearing gloves does not mean that you now lack the ability to transfer bacteria from one place to another. You need an education about removing gloves, periodically, so that they don't get worn out, they don't get holes in them, for example. And additionally, either removing gloves or washing them after you've touched, food surfaces. So with that story, let's jump into what we're going to talk about today. Again, the intrinsic factors. So what are some of the things that are inherent to foods, and how do we modify these to prevent, could even be for fermentation, to promote the, the growth of certain microorganisms? And particularly what we're going to focus in on today are three intrinsic factors: nutrients, pH, and redox. So nutrients, before we can talk about what do bacteria need when they're in a food and they're growing, well, we need to talk about, well, what exactly makes up a bacteria. So have on the right hand side, we have our little cutaway of a bacteria, what it looks like, from the inside. And in this list here is basically the makeup of a bacterial cell in terms of its dry weight. So we've gotten rid of all the water, and we've asked how many macro and micro and small molecules are making up this organism. We see the majority of a bacterial cell is actually protein, more than 50% of the dry weight. The next approximately 25% is made up of what we call the nucleic acids. So these are going to be DNA, RNA, so the things that make up the genetic material of the bacterial cell, and that allow the the processing of going from genes to making proteins. And it's also referred to as the nucleoid. So that section there in the middle of the cell. Phospholipids, so we talked about those making up the inner membrane, particularly. The lipopolysaccharide, which is making up the outer membrane for gram-negative bacteria, and the peptidoglycan layer. Again, it's going to vary depending upon whether you're gram-positive or gram-negative, how much of this is peptidoglycan. And now a bunch of smaller molecules, glycogen, which can be an energy store, some small molecules, vitamins, inorganic ions, all, of course, adding up to 100% of the dry weight. For the inorganic ions, the most common ones that, I'm going to say common, ones that are really critical for microbial growth include things such as magnesium, iron, sodium, potassium, and phosphate. Magnesium is a critical ion for a molecule that's called DNA polymerase, which, as the name implies, this is the enzyme that synthesizes a new piece of DNA from an existing piece. Obviously an absolutely critical enzyme for helping bacteria divide. So a very important ion. Bacteria also require low levels of iron in order to grow. And in fact, some of the ways that one bacteria can inhibit the growth of another one just naturally is they'll secrete proteins that are called siderophores, in which the role of them is to exclude the availability of iron from a neighboring bacteria. So iron, as we'll come back to a couple times this semester, is absolutely critical for all bacteria. Sodium and potassium are often used for just balancing, the ion concentration, the water, flow across a membrane. And phosphate is a major component of the DNA. So, DNA is a negatively charged molecule. The negative charge comes from the large amount of phosphate that's incorporated, into it. So let's look at, so all the things that a bacteria requires. And, so from a very basic standpoint, and again, this is fairly intuitive, foods that have a higher, amount of nutrients, but all other things being equal, are generally going to be better growth substrates for bacteria than are things that have a, very low, content. So taking these two liquids, into account, milk on the left, black tea on the right, we see that, with milk, there's, what we call the casein micelle. So this is where, some of the major, proteins of milk are found. Milk is, has about, 26 grams, of protein, per kilogram. We also have a fat globule, so supplying, again, a lot more nutrients. Looking down at the bottom, some carbohydrates, particularly lactose, which is the main, simple carbohydrate found in milk. Whole bunch of minerals, trace elements as well, bunch of vitamins. Basically, the story is not to memorize this list, but milk is a wonderful substrate. Not only for nutritious substrate for humans, it is a very, very wonderful medium for bacteria, to grow in as well. And clearly, there's a lot of nutrients there. Looking at black tea, which I think you can appreciate, doesn't support growth of bacteria very, very well. You can leave tea out at room temperature for a long period of time. It's going to last much longer than if you were to do the same thing with milk. There's only a little bit of protein, trace amount of lipids, a tiny, little bit of sugar. You do have some minerals. You will find some trace elements, but again, iron is almost undetectable in black tea. And as we mentioned in the previous slide, iron is one of those absolutely critical elements for bacteria to be able to grow. Over here, iron is found in fairly sufficient quantities in milk. And many of the vitamins, also are at very low or undetectable levels. So basically, if you're a bacteria, you want to grow in milk, you got a lot of ways to be able to do it. If you want to grow in black tea, you better be able to synthesize a lot of things, that you need, because there's not a lot of components that are, available in black tea. But it's not simply the presence of nutrients that can also limit the growth of bacteria. We also have to think about it's the accessibility of these nutrients as well. So coming back to our black tea example, we said there are some a low level of proteins that are found in teas. There are a lot of bacteria that can break down proteins into amino acids and generate energy from that. But in tea, a lot of the protein is in the insoluble form. So it's not dissolved in the aqueous environment, which is needed for the bacteria to be able to use it, because of the production of compounds such as tannins, in which the role of tannins is to basically bind proteins. So this is one way that bacterial growth is inhibited. Additionally, while you will find some carbohydrates present in tea, a lot of them are not available as monosaccharides and disaccharides in the form that bacteria like to use. They're in these larger polymers, such as cellulose and starch, in which most microorganisms don't have the ability to break these large molecules down into smaller molecules that they can then use for energy or for building blocks of a bacterial cell. So remember, it's not only the presence and absence of nutrients, it's also how accessible are they to microorganisms. Milk is actually another good example of something in which there are a large amount of nutrients, but again, they can be tied up in various ways. So we mentioned milk has a lot of protein, particularly in these caseins, but caseins, again, are large, proteins. Most bacteria, they can't do much with a protein. They need to be able to break a larger protein down into its individual amino acids. And it's those amino acids that they can use for, building blocks of other enzymes, for generating energy. So, if you're a bacteria that is going to be really successful at growing in milk, certainly you can use some of the free amino acids that are present in milk at a low level. But if you want to grow to really high levels, you're going to need to find a way of hydrolyzing these milk caseins down into amino acids, which is the form that you can really use. Additionally, we've mentioned that the most abundant carbohydrate found in milk is lactose. Milk is the only natural source of the carbohydrate lactose. So what does that mean if you're a bacteria? If you're a bacteria that's evolved to live in the environment, you naturally live in soils or similar places, you're never going to see lactose in your entire life. So you're probably not going to have evolved the ability to be able to use this carbohydrate as an energy source. You land in a vat of milk, you're not going to know what to do with lactose. So that might actually inhibit, or not inhibit, but prevent, your growth a little bit, as opposed to microorganisms that are more adapted to milk that have the machinery for being able to break lactose down into components for, building bacterial cells and for generating, energy. Okay, another, good example, of a microorganism when we're talking about, the limitation of nutrients and how organisms can get around this is Pseudomonas. In this particular example, Pseudomonas fluorescens. So what will become abundantly clear throughout this semester is there's a lot of different food spoilages that are food spoilage that's caused by the Pseudomonads. They are the most prominent spoilage organism in milk. Whenever milk is spoiled in the refrigerator or you've left it out, that awful odor, that you get, or some of the clotting that you get in the milk, a lot of that can be due to the Pseudomonads. But this is an organism that also naturally exists out in soils and in the general environment, and it doesn't have the ability to utilize lactose. So how does it cause such big issues with milk spoilage if it lacks this really critical characteristic? So there really are two different ways, and we'll revisit these when we start talking about food spoilage. Pseudomonads produce a number of enzymes that are called proteases that, by the general name, proteases break down proteins. Interestingly, these are incredibly heat-stable enzymes, so they survive a process called UHT, or ultra-high temperature pasteurization, that we usually use to try to preserve milk. These enzymes can break down the large milk caseins into smaller peptides, into amino acids, that now Pseudomonas can use as an energy source. Again, as when we talk about food spoilage, we'll also learn the ramifications of this is the generation of small peptides from caseins. These peptides are something that our tongues perceive as bitter. And additionally, the breakdown of amino acids that Pseudomonas can also do fairly well can generate ammonia, sulfides that are very characteristic of the spoiled milk perception. Additionally, Pseudomonas can produce a number of lipases. So again, by the name, these are going to liberate free fatty acids, away from mono, di, and triacylglycerides. The most common short-chain fatty acids, or SCFAs, that we find in milk are the 4-carbon and the 8-carbon that are often perceived, by our sensory, perception as being, rancid, being off flavors. They can react with other ketones and aldehydes that are found in milk to form, cardboard, flavors that are described as cardboard. Be esterified with ethanol that might be found in milk to form various fruity flavors. Again, we're going to revisit all of this when we come back to talking about spoilage organisms, particularly Pseudomonas, and the mechanisms by which all of these occur. But I really just wanted to use this slide as a way of pointing out that this is a way that some bacteria, or a great example of how bacteria can deal with the limited availability of nutrients in particular foods. Another really nice example of this is when looking at E. coli and its ability to grow on the surfaces of various produce, particularly romaine lettuce and spinach, in which have been a big problem with E. coli contamination in the past few years. If you may be aware, the last 3 falls around October and November, there have been large E. coli outbreaks associated with romaine lettuce in the California region. So there's been a lot of interest in trying to understand how this happens and how to prevent this. So this is information that was taken from this research article down here that you can look up if you were interested. But it's kind of pointing out what was a little bit of a mystery for a while about how does E. coli actually grow on the surface of produce, because produce, the surface of it, you can appreciate it's dry. There's almost very few nutrients that are available there for the bacteria, but clearly we know from these outbreaks that E. coli is somehow persisting, in these, it seemed to be inhospitable, environments. One of the explanations for this that these researchers found is that E. coli by itself, so we're looking at this graph, we're looking at the survival of various bacteria. So log CFU per gram of leaf, so basically the abundance of the bacteria over time. So they basically were taking these bacteria, they were inoculating them onto the surfaces of lettuce leaves and following survival over time. And as I was just saying, lettuce leaves are not a great environment for E. coli. About 50 hours after inoculation, it started to die off, basically, to levels very close to what the initial inoculum was. But what we see with these experiments up here, where E. coli is able to grow to higher levels, in here, these examples, E. coli was put onto the lettuce leaf along with another organism, such as Dickia dendanii, which is part of a group of bacteria that includes several Pseudomonads, some Erwinia, Pectobacterium, Xanthomonas, which are organisms that naturally exist or can exist on the surface of lettuce. And they have some, they are not human pathogens, in any way, shape, or form, but they produce enzymes that actually break down the cellular structure of lettuce, similar to what you might see here. And when you do that, you open up, a lot of the cellular material that makes up the structure of lettuce. And when you break open these cells, of course, you're releasing nutrients. So there's not a lot of nutrients on the surface of a lettuce leaf, but add E. coli along with a bacteria that's able to break down, the cellular structure, and E. coli is now able to grow using those liberated nutrients. So again, a really neat example for how co-inoculation can actually promote the growth of various foodborne pathogens such as E. coli in these environments. Okay, so that's all I really want to say about nutrients, and how they impact growth and survival of bacteria. The next intrinsic factor we'll talk about is pH, which, again, as defined, in previous classes that you had taken, that pH is basically the inverse concentration of, protons. Put another way, at a pH, when an environment has a pH of 2, that means the concentration of free protons is 10 to the minus 2 molar, 10 to the minus 13 when the pH is 13. We'd also mentioned in the first lecture, based upon the preferred pH, that we can define microorganisms into one of 3 categories. And we had mentioned that alkalophiles are something we really don't discuss too much in food microbiology. And again, here's why. We said egg whites, fermented shark are really the only 2 examples of alkaline foods that are out there. And it mentioned that most foods tend to be around neutral to somewhere in the the acidic region. So pointing out that foods such as fish, milk, for example, they tend to be spoil fairly well because the pH is near neutrality, where a lot of our spoilage organisms really prefer to grow. Citrus fruits, soft drinks, things that have lower pHs, and again, I think just from experience, you kind of appreciate that these are foods that don't tend to spoil very quickly. So from the standpoint of how do we apply this knowledge to food microbiology, so here's one of a couple of charts I'm going to put up on the screen for you for the first couple of lectures, when we're trying to just discuss what are some of the various guidelines for trying to understand whether a particular food is going to be susceptible to microbial growth. So the word general is in red and underlined on this table for a reason, because these are not absolutes, but these are general guidelines if the only thing that you know about a food is its pH. And you're trying to gather some understanding of, is this supportive of growth of bacteria, of yeast, of molds, of all three, or of none? So for bacteria, as we mentioned, most bacteria, spoilage bacteria and pathogens, they really prefer to grow around neutrality, maybe just a clip of, above or below. In general, once a food gets below a pH of 4.5, at least in terms of, foodborne pathogens, it's not going to support the growth of bacteria anymore. And likewise, when the pH gets up to the range of about 8.5 to 9, same thing. So a couple of things. So again, these are general, rules of thumb. These are not absolute. There are bacteria, for example, that can grow at a pH of less than 4.5. But again, these are kind of general, guidelines that we use in food microbiology. The second thing, and this is really, really important, I can't emphasize this enough, because I think this is one of the things that I find that a good part of the class often doesn't appreciate. Remember that these are the values for growth. These are not values for survival. So if you take a bacteria that, say, can't grow, below a pH of 4.5, and you put it in a food that's pH of 4, that means it's not going to grow. That bacteria might be able to survive, though. So it's still living, it's still there, it's just not replicating. And if it finds itself in an environment that is, more conducive from a pH, range, it's now going to be able to grow again. So remember, these are values for growth, not for survival. What the take-home point that you see with yeast and molds is, in general, they have a broader range of minimum and maximum of for growth than do bacteria. Both yeast and molds, generally somewhere in the order of about a pH of 1.5 to 3.5 is their minimum cutoff for growth. Most of them prefer acidic to slightly acidic pHs for maximum growth. And for yeast, they can probably, most of them can grow in foods up to about a pH of 8.5. And molds, again, are, the ones that are superior here. They can grow, generally grow up to a pH of about 11. So, and I put up a couple of slides, previously, and said, hey, this is kind of just more for to be able to conceptualize. Don't memorize it. This is different. This is one of those tables that you really need to know these values off the top of your head, because there will be examples of things that I'll give you in which, here are some of the various characteristics of this food. What predictions would you make in terms of bacterial, yeast, and mold growth? Okay. Good. Okay, so another concept, that we need to talk about when discussing pH is that sometimes it's not, the effectiveness of a food at being able to prevent the growth of a bacteria or even being able to kill it is not only the pH, but also what other weak acids might be present in that food, and how does that relate to the pH. So let's start up here with this table that was taken right out of a food microbiology textbook. So you hopefully appreciate, remember from your biochemistry classes that there's a whole bunch of acids that fall under the heading of weak acids. And what that basically means is these are acids that can exist in one of two different states. They can either exist in what's called the protonated form, so this is when, they're at a lower pH and it had the proton is associated, with the acid, or at a higher pH in which the proton and the acid are what we call dissociated. Good example over here is acetic acid. So we have basically what they call the abundance of the associated form or the dissociated form of acetic acid. As you raise the pH of a solution, the concentration of the dissociated form increases, the dissociated form, the concentration of the associated form decreases. And for all weak acids, there's going to be an inflection point right in the middle, which is that pH in which you have equal concentrations of these two different forms. And that pH is what we refer to as the pKa. Some acids, like acetic acid, that only have one group that can be dissociated, they're just going to have one pKa. If you take a look at a molecule like citric acid, there are 3 different carboxylic acid groups associated with it, so there are going to be 3 distinct pKas. So how does this relate to antimicrobial activity? Well, here's the really important take-home point from this. So as we just mentioned, by changing the pH, you can change the ratio of the associated and the dissociated form. And remember back to what we were discussing in the last lecture, that the cytoplasmic membrane of a bacteria, gram-positive or gram-negative, is only permeable to hydrophobic small molecules. And really, I don't want to say hydrophobic, but really uncharged small molecules. And water is the other one. So what that means is, in this form, acetic acid, it's a small molecule, but it's got a negative charge, is not going to cross a bacterial membrane very readily. But in this form, it's now a small molecule, it's uncharged, it's going to cross the membrane a lot easier. So let's take this schematic down here. Again, we've got the bacterial cell that's shown here in this outline. We have some theoretical weak acid that's on the outside of the bacterial cell. Again, in the dissociated form, it's not going to cross the membrane, but in the associated form, it will. So when do we have higher concentrations of this form? But it's when the pH is at a pKa that is it's when the pH is much less than the pKa of that weak acid. So in that case, the associated form is now free to cross the membrane and come into the cytoplasmic environment of the bacterial cell. So obviously the outside is going to be a low pH. That's how we got the acid into its associated form. Once it crosses in the bacterial cell, the cytoplasmic space is generally going to be neutral, maybe slightly acidic, a pH of 6.5 or so. So now we've gone to a pH in which we've encouraged dissociation of the weak acid, as shown over here, being this reaction being driven to the right. So the acid dissociates. What that does is it increases the concentration of free protons inside the bacterial cell. As we mentioned before, what's the definition of pH? But the concentration of free protons that are available. So what basically happens as the weak acid comes across the membrane, it dissociates, it starts to drop the pH inside the bacterial cell. The bacteria hate this, of course, because they really want their cytoplasm to be neutral or maybe slightly acidic. That's where their enzymes work best. That's where they really have the most robust physiology. So the bacteria, they need to find a way of getting rid of these free protons. So what they do is they pump the free proton outside of the cell, and they'll do it using an enzyme that requires them to hydrolyze ATP back into ADP. So putting this all into action, if you have a weak acid outside the bacterial cell, a pH that is much lower than the pKa, you'll get a lot of the weak acid going in, dissociating, and the bacteria is spending a lot of its energy, a lot of its ATP, to constantly pump these protons out of the cell in order to keep their cytoplasm around a preferred pH. So the bacteria is spending a lot of time, trying to keep it, this neutral cytoplasmic membrane. It doesn't have a lot of energy for doing other things. and eventually this can overwhelm the entire bacterial system. So this is a really important concept because we use weak acids a fair amount in food preparation. They're naturally present in certain foods. For example, citric acid is the acidic perception that you get from a lot of citrus fruits, for example. And how antimicrobial those weak acids are has everything to do with what the pH of that particular food is. So that, that's a real important concept to keep in mind. So the last intrinsic factor I want to talk about is redox, also abbreviated as E with a subscript H. A little bit harder of a concept to wrap your head around, but we're going to try to do a little bit of some mind tricks to try to help you with this if this is the first time you're hearing about this. Redox is probably something that was covered in organic chemistry, and it basically is the concept when you have a compound that can exist both in an oxidized and a reduced form. NAD, NADH that we talked about before as an example of such a compound. That there's going to be some tendency for it to exist in, or a certain percentage is going to exist in the oxidized form. A certain percentage is going to exist in the reduced form. In your chemistry classes, it would have defined redox as the tendency of a media to accept or donate, electrons. May or may not be a very helpful definition to you. One thing that really isn't terribly helpful is if you look up the formula of redox, which we're basically saying the redox of something has to do with the standard redox potential, the gas constant, the temperature, the number of electrons transferred, the Faraday constant, and the natural log of the concentration of the oxidants and the reductants. Really nice if you're trying to use math to make some calculation about your environment. Again, not exactly, to me at least, the most useful way of of trying to understand what redox is. I look at it this way, and this is what really helps me with this concept, is redox has a lot of similarities to pH. So look at it this way. When you learned about pH, you learned, again, that pH, and again, coming back to our weak acid example, is the pKa of an acid plus the ratio of the, basically the dissociated form, so the protons and the acid over the concentration of the associated form. So it's a way for you to be able to calculate what the pH of a solution is based upon the pKa and the ratio between these two forms. The formula for redox actually looks very similar to with the formula for pH if you think about it. So we have redox instead of pH. You have your the redox potential here, which is similar to the pKa, and then basically plus the concentration of the two different forms. So the dissociated form, which is similar to that guy there, and the associated form, or the protonated form, which is that one there. So I really think of it more as kind of an equivalent to pH, but this time we're looking at oxidation and reduction rather than acidic or basic conditions. So let's talk about, but really, how does this relate from a practical standpoint for food microbiology? So it has to do partially, or how this impacts the redox of a food. So we've got a couple of examples on this slide of various oxidant reductant pairs. So again, this is analogous of the two different forms of a weak acid. What the standard redox potential is for each of these, and also pointing out, so the redox potential of a food, it really has 6 things that it depends upon. The first is what oxidant and reductant pairs are present. Again, that should be pretty obvious. Depending upon what is present, that's going to dictate the value of redox, which we measure in millivolts, the ratio of the oxidants to the reductants. Again, we can see that from this formula up here. Again, change the concentration of oxidants and reductants, and it's going to change ultimately, what the redox in that particular environment is. Interestingly, you can change redox with pH. So for every 1 unit of pH, or a decrease in pH, you're actually going to increase the redox by 58 millivolts. You can actually kind of tell that from this formula. Again, as the pH goes down, the concentration of free protons is going to go up, and that would increase, ultimately your redox. What's called the Poisson capacity. So again, that's going to be analogous to the buffering capacity of pH that we associate with pH. So basically, as you add more oxidants and reductants, how much does that environment resist change? Kind of like buffering, in which, buffering is the idea of, as you add acid or base to an environment, how much does it resist, changing pH? Number 5 is a really big one. So the availability of oxygen that's available in the food or inside the packaging material. Obviously, the oxygen, water, ratio has a very high, redox. And additionally, oxygen is going to be present, in most foods in a very high concentration. So what you find is foods that are stored outside in the air, a lot of the redox can actually be driven to a high positive level because of the concentration of oxygen that's present. Additionally, microbial activity is a big factor. For example, as you ferment a food, the redox is typically driven downward. Part of that is because you're going to generate a lot of reduced compounds such as NADH that will drive the the redox downward. So anytime you're looking at a fermented food, if there's a lot of microbial activity in it or such, generally that's going to be a way of decreasing redox. This guy we'll bring up on the next slide. So glutathione is this compound right here that can be oxidized or reduced by that thiol group that's shown in yellow. So let's take all that now and put it into, again, a kind of a practical discussion using some actual foods. So if we look at the redox values that are found, and we're going to pair these off by common foods here. So if we have raw meat, post rigor, so this is freshly slaughtered meat that's been allowed to undergo the rigor mortis process, which is basically the meat utilizing a lot of the remaining glucose or carbohydrates that are present in the muscle tissue. Like with microbial activity that we just mentioned, metabolic activity will tend to drive the redox down. Again, you're producing a lot of reducing components in that process. However, you take that meat, you now grind it up, you mince it, you incorporate a lot of oxygen, and now the redox is going to come to a more positive level. Take that or something similar, like sausages, canned meats, and now you cook them. Cooking process is going to drive off a lot of oxygen, and now you'll take something that has a positive value and you'll reduce it down. Some various grains, such as wheat, will tend to have a really low, redox potential, which at first seems kind of counterintuitive. I mean, it's something that is generally stored, in kind of the open air environment, but pointing out that wheat has a very high concentration of glutathione. So it has a, kind of a pair that will tend to drive, the redox lower. Barley, kind of being the opposite. So a grain that is generally stored, again, in atmospheric concentrations of oxygen, doesn't have a lot of glutathione. The redox is going to tend to be more positive. Potatoes, I think we kind of appreciate leaving potatoes sitting around. There's a lot of metabolic activity going on in which eventually that potato will start to, sprout roots. That metabolic activity is going to give it a lower, redox. Higher redox just for some general, fruits and vegetables and such. So from a standpoint of trying to understand, microbial growth in various foods, again, if the only thing that you know about this food is it's redox, and again, pointing out that these are preferred redoxes for growth. These are not required for survival, just for growth. For strict aerobic organisms, our spoilage organisms such as Pseudomonas fluorescens, generally going to want a redox between about plus 100 to plus 500 for optimal growth. Yeasts and molds are organisms that usually are going to require high levels of oxygen for optimal growth. Again, they're going to want a higher redox as well. The redox goes down a little bit for the facultative anaerobes. So these are organisms that generally can grow in lower quantities of oxygen, and again lower redoxes for the anaerobes. So it tends almost to kind of segregate by oxygen requirements. For reasons that we've already mentioned. But what I do want to make, hope is abundantly clear here, is redox is distinct from oxygen availability. You can tell that with the anaerobes. Some anaerobes can grow even at positive, redox values. So you can have positive redox values in environments in which there's no, oxygen present. So that was, kind of our discussion of, three very intrinsic factors and how they dictate microbial growth. And we'll kind of end it again with this example here of how we can change the intrinsic factors of a food to slow down or more minimize the growth of various bacteria. So again, coming back to our example here of milk, we know that there's a lot of nutrients that are present. It's got a pH near neutrality, a little bit underneath. Has very high what we call water activity that we'll address in the next lecture, and it has a positive redox. A lot of things that are conducive for microbial growth, and we know that even refrigerated, some milks, are generally going to last, maybe, probably 7 days, maybe, today with improvements in pasteurization, even up to 2 weeks. But again, it's something that readily spoils. Now we take buttermilk, which if you compare it side by side, it has really almost the same level of nutrients, and definitely still has a very high, or has an abundance of nutrients. Also has a high water activity, positive redox, but the one difference is the pH of buttermilk is going to be about 2 pH units, less than that of fresh milk because you have fermented it. You basically have taken milk, you've added lactic acid bacteria such as Lactococcus lactis that have grown, have converted the lactose into lactic acid, dropped the pH, and just by changing that one intrinsic factor, we now have something that probably has a shelf life of about double that of fresh milk. So a really nice example of how changing an intrinsic factor in a food can be used to preserve it. So that is all we're going to talk about today. So there will be a short quiz for you to take right after after closing down this video, and I'll stop sharing my screen. We're kind of get used to this, so take the quiz, and all the information that we've talked about so far is what we will cover in our first face-to-face discussion on next Wednesday. So hope that was good, and we will just catch up with you in the next lecture, in which we're going to continue talking about some of these various intrinsic factors. So take care.